Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Accelerating Cell and Gene Therapy Success with cGMP Gene Editing Components and Regulatory Compliance. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout this presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank GenScript who developed the content for this presentation. GenScript is a leading contract research organization in the world providing gene, peptide, protein, CRISPR, and antibody. Since its foundation in 2002, GenScript has grown exponentially through partnerships with scientists conducting fundamental life science research, translational biomedical research, and early-stage pharmaceutical development. The company is recognized as having built a best-in-class capacity and capability for biological research services, encompassing gene synthesis, peptide synthesis, custom antibody and protein engineering, and in vitro and in vivo pharmacology, all with the goal to make research easy. Now it's my pleasure to introduce your speaker for today's webinar, and he's Dr. Jianpeng Wang. He is the Senior Director of GMP Manufacturing at GenScript and a seasoned professional with a PhD in chemistry and more than 15 years of experience in early R&D, process development, and GMP manufacturing of nucleic acids for gene editing. Under his leadership, Dr. Wang's Matrix team has delivered more than 100 batches of cGMP products to support over 40 projects globally. Now it's my pleasure to pass the mic and the controls over to Dr. Wang. So Dr. Wang, when you're ready, you may begin.
Thank you for the kind introduction. Hello, everyone. I am Wang Jianpeng from GenScript. I am the Senior Director of the GMP Manufacturing, and so glad to be here, have this opportunity to have this sharing. And the title for today's presentation would be Accelerating Cell and Gene Therapy Success with cGMP Gene Editing Components and Regulatory Compliance. And today's talk will be divided into 4 parts. First, I'm going to introduce about GenScript's gene editing portfolio. And second, I'm going to share with the evolving regulatory landscape. And after that, we'll have some case studies that happened during our delivery history. And the last section is about the Q&A. Okay, let's dive into the presentation here. First is about the gene editing portfolio introduction. So GenScript is one of the leading vendors for the nucleic acids raw materials for CRISPR in the world. And we have the Gen CRISPR solutions, mainly supporting the non-viral CAR T and the TCR-T cell therapy development work. We have a bunch of products, but today I'm going to focus mainly on 2 parts. First is about the CRISPR components for the knockout application, mainly for the single-guide RNA, Cas9 protein, and ESP Cas9 mRNA. And the second part is about the DNA knock-in templates. It is HDR knock-in templates. We have different modalities for that, mainly the gene exact linear single-strand DNA, gene wand linear double-strand DNA, and also gene circle plasmid double-strand DNA. And today's topic will be further centralized on the 4 products, the single-guide RNA, and also all the modalities of knocking DNA templates. So speaking of the RNP-based CRISPR knockouts, so the workflow is very straightforward. The chemically synthesized guide RNA will be incubated with the Cas protein to generate the RNP formation, and this RNP will be introduced to the cells for the knockout applications. And the single guide RNA is chemically synthesized with some chemical modifications. And the advantage of using this chemically synthesized guide RNA is listed below. So first of all, it is a non-viral delivery system. It is DNA-free, so it will bypass the transcription and the translation processes. And it has the highest editing efficiency with minimal off-target rates, and of course, the lowest cytotoxicities. And for the RNP-based CRISPR knock-in, the workflow is also very straightforward. So after the formation of the RNP complex and the HDR knock-in templates is also going to be designed, manufactured, and introduced to the cells together with the RNP for electroporation. And the cells is further studied for the downstream works. And the advantage of using this HDR templates is also listed here. First of all, it is a non-viral delivery system, and we provide some versatile modalities to fit into different scenarios and different cell types. And with these different modalities, we have improved the knock-in efficiency and the low cytotoxicity. And our products are designed to be suitable for scaling up. Thus, it paves the way for the future CMC work, and it is very suitable for GMP manufacturing. And here is the detailed format of the three modalities for the DNA knock-in templates. First of all, this single-strand DNA, it is called the gene exact. It has a minimal cytotoxicity for highly efficient gene insertion. And the length of this modality can be reached up to 5 kb. And this is the gene 1, the double-strand DNA. You can see it has a close-end structure. Thus, the stability has been greatly improved. It is very suitable for the long gene knock-in at a large scale. And the length of this modality can be reached to 10 kb. And this is a new product we recently launched. It is a gene circle double-strand DNA. It is a plasmid-like double-strand DNA with a very small amount of backbone sequences. We have only about 430 base pair in the backbone. And this plasmid double-strand DNA has no antibiotic resistance or any CpG islands. So it is a very unique modality that's suitable for example, transposon-based technologies. And the length of this modality can be reached up to 20 kilo BP. And of course, all of the three modalities, we have the manufacturing capability from the discovery phase all the way to the cGMP manufacturing phase. And this is our cGMP manufacturing capability introduction. We have a newly expanded facility that is in use starting from last year, and we have started to deliver the cGMP batches in this plant starting from last August. And this is a manufacturing space, greater than 400,000 square feet. And we have multiple independent facilities with segregated greasy manufacturing rooms. And of course, in this plant, we can produce sterile products. So in the grade C manufacturing room, we equipped it with some grade A isolators for sterile filtration and the aseptic fill finish part. So with all the hardwares and workflows, we also have our fully integrated quality management system to support the compliance and of the manufacturing process. And to support the IND filing, we have a very comprehensive QA and QC documentation system to support that. So in this plant, we have 8 single-guide RNA manufacturing lines that can support tens of milligrams to gram level of GMP manufacturing. And we have 4 manufacturing lines for linear single-strand DNA or double-strand DNA, which we can fit into up to 100 milligrams of GMP scale. And we have 2 manufacturing lines for gene circle manufacturing, and it has up to gram level of cGMP manufacturing. And you may wonder what's the difference of the manufacturing between RUO, which is non-GMP, to cGMP. So the most important thing, speaking from the facility, is the segregation. So for the RUO manufacturing, we will adopt the ballroom concept. So all the instruments will be put in a ballroom, no matter for the synthesis or for the purification, all the instruments will put in a ballroom. However, if it is in the cGMP manufacturing, all the rooms are segregated according to each manufacturing step. And if the purification is going to be conducted in a clean room, it is in greasy area. And only GMP products will need the sterile property. So the grade A isolator in the greasy background is designed to support the aseptic fill finish part. And speaking of the capacity and the average lead time or the scale, the RUO and the GMP differs significantly. So there's only one sequence manufactured in the workshop each time. And this is the more detailed introduction of the 3 products: the single-guide RNA, the linear single-strand or double-strand DNA, and the gene cycle. So the upstream of the manufacturing process will be conducted in a controlled but non-classified, which is CNC area, including the chemical synthesis of the single-guide RNA or the upstream preparation of the linear single-strand or double-strand DNA or the cell banking for the gene cycle. And most of the purification part are conducted in greasy area. For example, the purification of single-guide RNA and also the purification for knock-in templates. And the final step, which is the sterile step, will conducted in the gray A area in greasy background. So mainly the sterile filtration and the aseptic fill finish part are conducted here. So this is the general manufacturing workflow for the 3 products. And we have delivered over 120 clinical batches till last month, and we have over 30 successful global audits, and we have supported over 50 IND filing projects, and 9 of them have received global IND approvals. So to achieve this, we have seamless RUO to GMP transition to accelerate the process. So for the RUO phase, mainly it is designed for discovery purpose. It is mainly for screening of gene of interest, the in vitro functional assays, or some small-scale in vivo validations. So we will adopt the ISO 9000 quality management system in this phase. When it moves to preclinical development work, which is the animal study or the tox batch, we can adopt GMP-like or cGMP quality management system here. And when it comes to the IND filing, and after that is the clinical trials or even the commercialization manufacturing, we will have the cGMP quality management system to support this. And our GMP quality management system are designed based on the ICH and FDA system-based compliance framework. So the 6 subsystems are strictly followed according to our quality management system, which is a quality assurance, quality control, production, facility and equipment, material, and of course, organization and personnel. And to support our clients from the early phase to the late phase, we will adopt some flexibility, especially at the preclinical study. So we will use these phase-appropriate quality management systems, and some flexibility are allowed at the early phases. However, when it comes to IND filing and the clinical phases, we will require more and more validation work, and we will put more and more process controls. For example, the analytical methods and the manufacturing process need to be validated before the commercialization part. So with this quality management system in place, we can significantly reduce the quality cost. So overall, we can achieve the cost-effective manufacturing to support our clients. Okay, the second part is about evolving regulatory landscape. So the first CRISPR-based therapy was approved by FDA last year in December. It is from Vertex, and it is designed for the sickle cell disease. And during the evolving of the FDA regulation, the most important concern with the cell and gene therapy is about the potential of target editing effect. And also, we need to assess the serious risk of secondary malignancies and the off-target effects during the long-term safety observation. And US FDA guidance has specified the design, manufacture, and the testing of the CGT products. So it is evolving, and the regulations will be continuously updated and refined based on the evolving understanding and the experience with the CGT product approval processes. So FDA encourage sponsors to initiate communications even from the early stage for product development with FDA. So this is the main regulatory landscape for the CRISPR-based therapy. And the regulatory considerations for the CRISPR-based gene editing components are mainly due to the potential of target genome modification. So the genome editing component manufacture and the testing will be divided into mainly three categories due to their different application scenarios. Taking single-guide RNA, for example. So if it is used in the in vivo scenario, for example, if it is encapsulated together with the mRNA in an LNP format and injected into human beings, it is considered as drug substance. And if it comes to the ex vivo application, for example, the UCAR-T, the universal CAR-T, the cells isolated from the patients will be treated by the CRISPR technology, the RNP delivery format mainly. And in this scenario, the single-guide RNA will be considered as critical components. And if it comes to the iPSC scenario, because the iPSC requires a master cell bank, so the single-guide RNA in this scenario is considered as a starting materials. So no matter in which scenario is the sgRNA, FDA requires the sponsors to put more efforts and to more attention to the following aspects. First, need to evaluate the sterility, identity, purity, and activity of the gene editing components. And we need to pay attention to the process residues. And of course, the analytical methods utilized for the gene editing components should be qualified. And we should put some in-process controls during the manufacturing step. And the stability study is also very important, especially for the long-term stability. And the phase-appropriate GMP quality management system is adopted because it is an evolving process. And we need to have adequate facilities and manufacturing operations to prevent any potential cross-contaminations. So let's dive into the specific requirements, and also we take single-guide RNA as an example. FDA has evolving the considerations for single-guide RNA, taking the purity, for example. So normally, single-guide RNA purity, now it requires greater than 80% for the full-length product, which is FLP. And recently, it requires that any impurities that is greater than 100% should be identified. So this is about the purity. And also for the sequencing confirmation, the next generation sequencing is recommended to be used as the identification method to ensure the correct full-length sgRNA sequence is present. And also, there is a merging requirement that requires using NGS as an orthogonal purity assay to identify and quantify the presence of the sequence variants. And this is for the sequence confirmation and orthogonal purity test. Due to the safety concerns, the residue solvents and the elemental impurities during the manufacturing process should also be accessed. And the stability, especially at the recommended storage temperature, with the long-term stability is needed to observe the long-term effect. So this is the FDA's evolving consideration for single-guide RNA. And what GenScript can do to support the client's regulatory requirements. So we can do at different stages. So for the preclinical stages, our scientists will work closely with your scientists for some gene editing designs. We have some online tools for the sequence design, and we can also hold some technical discussions about the project design. And when it comes to the IND filing, our scientists can attend or support the pre-IND meeting with FDA. And we are compiling all the IND submission documents in a CTD format. And for single-guide RNA, we have submitted a DMF, a drug master file document that you can directly refer to that. And of course, we will respond to any FDA comments. When you pass the IND and move to the clinical trials, we, GenScript, as the manufacturer, will start to consider about the CQA optimizations and considering the scaling up for future cost saving. And if there's a major CMC change, we will submit any amendments document to FDA. And when it comes to the BLA phase, we will help to submit the BLA documentation, and we will respond to FDA comments. So two key points summarized here. There is no regulation, never been ahead of science. So scientists are willing to communicate with regulatory agency for better understanding of the quality attributes, as well as the impurity profile of gene editing components. And for single-guide RNA, we have submitted a DNA file, so you can directly refer to that. And this is also a confirmation for GenScript's platform process for manufacturing. It is in a consistent way. And don't worry about the change during that. The major CMC change is a joint work by GenScript and the sponsor. So we should work together to determine whether it is a change. So once the manufacturing change happened during the project, we GenScript will further evaluate whether it will has any potential impact on safety, identity, quality, purity, potency for phase 1, or any efficacy for phase 2 or phase 3 trials. If it is no, we will directly implement the manufacturing change. And if the answer is yes, we will align with the sponsor to see what kind of supporting data is needed to generate for the comparability work. And the CGT product manufacturing, which is the sponsor, when you receive the documentation, you will also initiate a risk assessment, and we will define the scope of comparability study, and we have several questions to be answered. Whether the CQA or CPP has been established, is the analytical studies sufficient, if the additional characterization work conclusive, whether any additional non-clinical PK/PD work conclusive. If the answer is yes, we will together conduct the comparability and the stability study, and the sponsor will submit for IND update. If the answer is no, so the sponsor will discuss with the FDA for the safety evaluation for phase 1 and the effectiveness for phase 2 and phase 3. So this is the mainly workflow for the collaboration for GenScript and the sponsors. Okay, so let's move to some real case studies. So first, the case study is about using NGS as the identification test. So NGS is a powerful analytical method to determine the sequence correctness. And there are some side reactions will happen during the manufacturing process. Single guide RNA is chemically synthesized, and there will be some side reactions that cannot be avoided. And for example, some reaction will cause a G to A mutation. A G to A mutation is not that significant in terms of mass spec, because the molecular weight will have only a difference of about 1 Dalton. However, if this G to A mutation happen, it completely changes the base pairing system. We know A will pairing with T and G will pairing with C. If a G to A mutation happen, we will have a misunderstanding of the base pairing and cause the potential of target effects. And what's more difficult is that the traditional analytical methods, for example, the UPLC or the HPLC, cannot distinguish the G2A mutation because it's too small. The speaking of HPLC, the retention time and the peak shape will, almost be the same. When it comes to the mass spec, because there's only 1 Dalton difference can be hardly detected. However, if this G mutated to A, the potential for off-target effects is significantly increased. And we have a, IGAD bioinformatic prediction for these two sites. If the G2A happen, we can see the off-target effects significantly improved. And the NGS can help us to identify the G2A mutation, and we will upgrade our manufacturing process for better quality control. So this is our NGS library preparation workflow, and this is our reproducibility test for the spiking G2A sample testing. And after confirming the NGS is powerful enough, we will do some, row tests. We will overkill this side reaction to confirm that the G will change to A. So the orange line is an overkill reaction. We overkill all the side reactions, and we can obviously see the G has changed to A. And when we optimize the manufacturing process, we can see all the red lines, all the green lines, we have completely eliminated the G2A mutation. So NGS here function as the analytical method is powerful or is more powerful than mass spec to tell the G2A difference and guide us to upgrade our manufacturing process. And the second part is using NGS for orthogonal purity testing. Based on the previous knowledge of NGS, we further improved the NGS analysis workflow. We installed the UMI to the library preparation part, and using this technology, we can differentiate more powerfully for the N-1, N+1, and any point mutation analysis. And this is a showcase of how we do the NGS purity test. We can see the full-length product analyzed by NGS purity test is around 85%. And for all the potential sequence variant listed here, it has different abundance as low as 0.5%. And the FDA has required the impurity greater than 1% to be reported. You can see almost every one of them can be distinguished and identified by NGS as a purity test. And here is how we harnessing this NGS orthogonal purity testing for impurity profiling. So this is a real case. We worked with our clients and we blacked out all the guided regions in that. And we find that using NGS, we detect some mutations. We have base mutations there, and we have the N+1 mutation at different abundance here. And we using the in silico prediction to see whether this mutation will bring harm or bring any off-target potential to the gene editing experiments. We will see that this A to U and this U to A mutation has a higher potential to cause the off-target effects. And based on that, we will also do some wet lab test. So we de novo synthesized all the potential, mutations together with the FLP and the positive controls. And we will do the on-target efficiency test. We can see from this entry, the two mutations can cause significant, on-target efficiency reduce, which is, increased the off-target potential. So it is considered as a critical impurities. So we further spike in the products, the de novo synthesized products, together with the FLP at different scales and at different ratios to see how important or how severe it is as a critical impurity. We can see after spiking different ratios of the impurity, we will observe different editing efficiency of the target sites, and even spiked at a ratio of 50%. This A to U mutation will cause only about less than 4% of target. However, when it comes to this U to A mutation, if spiking 50%, we can observe, almost 70% of off-target effects. So this NGS as the purity, orthogonal purity testing method is also very powerful. It can help us to identify the potential impurities, and using this tool, we can further analyze whether the impurity is critical or not, and further guide to improve our manufacturing process. And this is another case study how we do the residual single-guide RNA detection. The USP, the US Pharmacopoeia has required the has required to detect the residual RNAs in the system. And besides the traditional HPLC and the mass spec analytical method of single-guide RNA for the long-term stability, we also developed the qPCR-based method to analyze the residual sgRNA even in the cell system. So the qPCR works like this. So there is a TechMan probe, one end with a fluorophore and one end with a quencher. It will bind to the sgRNA sequence. And if the fluorophore is cleaved from the quencher, it will generate the fluorescence signal. And we will utilize this fluorescence signal to detect the residue level of the single-guide RNA. And the pharmacopoeia requires the accuracy has, extraction range from 50% to 150%, and the RSD is required to be less than 30%. And in our testing of the LOD experiment, we can find that we can easily meet both for the 50% to 150% requirement, as well as this less than 30% requirement. So the RSD is set as 2.5 picogram. So we can detect single-guide RNA as low as 2.5 picogram. So we adopt this analytical method to the T cell test, and we can see we introduced single-guide RNA to the T cell system, and we also include the RNP, which is a single-guide RNA with Cas9 protein, to the system. And uh, both groups has a significant sgRNA residue level decrease, and they will decrease to a plateau after 48 hours. Because the Cas protein can sometimes, not easily stabilize the single-guide RNA, so it is more stable. We can see that the residue sgRNA is more than that of pure guide RNA in the cell system. And this analytical method helps us to determine the residual single-guide RNA in the system, and this can be further applied for any safety evaluation or assessment in the clinical trials. And the second study, the fourth case study is about the risk assessment. So risk assessment shall be, defined or designed together between the manufacturer and the sponsor. So this is a case of showing how we do risk assessment together with our client. So this is the HPLC HPLC trace for single-guide RNA. So there were existing some impurities. We categorized as the early eluents and the late eluents, and of course we have the MAMPIC. So to investigate which eluent is more harmful to the cells or to the editing efficiencies, we collected the eluents during the purification step, and we send the eluents to our clients, and the clients will perform a risk assessment test with all the eluents. So they will perform a mock group, and the early eluent and the MAMPIC and also the late eluent. They will evaluate the cell viability as well as the editing efficiency. And with all of this data, we confirmed that, the early eluent is not that favorable during the purification process. So we're tuning our purification protocol, mainly the mobile phase, the gradients, and to try to eliminate or reduce the early eluents. So this is the risk assessment determined together with GenScript and the clients. And this is case study 5 for how we determine to adopt the cost-effective control during the early phases. This is the iPSC-based manufacturing process, and we know single-guide RNA is a critical material used as the early manufacturing step of a CAR iPSC allogenic therapy, and the iPSC has a very long manufacturing process, and it has a C cell, it has a mast cell bank, and then come to the drug products. Single-guide RNA is used here together with the protein and the donor DNA. So it is considered as the raw material. Thus, we can design or use our GMP-like quality management system, which means we will do the segregate, clean room, EM, the environment monitoring program for facility, and we will have the manufacturing controls for the MBR, for sterility control, for cleaning and sanitization per SOP, blah, blah. And also we will have certain requirements for equipment, analytical methods, and we will, have the quality events resource invested during this manufacturing process. So this is a case study also together designed by GenScript and the customer to determine a cost-effective manufacturing workflow for iPSC-based application. And I hope all of the case study here have showed our understanding about the regulatory requirements. So we have CRISPR single-guide RNA, and we have the DNA payload templates, and we have the global filing supports, and all the detailed information listed here. We have, DMF for single-guide RNA, and a total 9 IND approvals worldwide. And we have a very dedicated and expertized team of QA, QC, and RNA, and also the project management team to support the global IND filing. So start your cGMP batch production with GenScript right away. So I think that's mainly about the introduction, and I'm going to share with you today. And the next, I'm happy to move to the Q&A section.
Okay, thanks very much, Dr. Wong, for the very informative presentation. Just a quick introduction for myself. My name is Scott Pritchett, and I'm a Global Market Development Manager here at GenScript. We do have a couple questions that were submitted during the talk, so I think we'll take a few minutes now to get to those. However, if anyone in the audience has further questions you would like us to address, just feel free to reach out to us at crispr@genscript.com. So with that being said, let's get started with a short Q&A. Okay, Dr. Wong, first question I have for you. Can you explain the advantages of using next generation sequencing over mass spec or HPLC for impurity profiling?
Oh, yeah. Thanks for the question. So first of all, impurity profiling is very important because it allows us to distinguish all the potential side products and the impurity species besides the target products. For example, the N-1, N+1, and any sequence substitutions for single-guide RNA. Because single-guide RNA is chemically synthesized and has a relatively large molecular weight, which is around 32 to 37 kilodaltons, it is very difficult for the conventional analytical instruments to analyze the side products and the target products. For example, the N-1 and the full-length product of single-guide RNA cannot be fully separated on HPLC. However, NGS is more powerful. It allows us to do more precise analysis about these impurities with the target sequences. It allows us to know the percentage and the presence, of course, of the N-1, N+1, and any sequence variants in the products. With these analytical methods, we can better know about the components and composition of the single-guide RNA, and further to control the manufacturing process to reduce or even eliminate the impurities.
Great, thanks very much. Okay, another question we have here is how does residual sgRNA and impurities affect the editing efficiency or off-target effects?
Okay, so the editing efficiency and the off-target effects are affected by several reasons. For example, the single-guide RNA sequence design, the Cas protein version, and also the ratio of single-guide RNA to Cas protein and the free single-guide RNA or free Cas system in the free Cas9 in the system, or the quality of single-guide RNA. Both the single-guide RNA and the impurities will affect the editing efficiency and the off-target simultaneously. So there is a dynamic balance for the equation. And according to our study, when the amount of single-guide RNA is less than a certain threshold, the editing efficiency and the off-target effects will be significantly reduced or even eliminated. Given the fact that normally single-guide RNA is greater than 80% in the product, so it contributes more to both the editing efficiency and the off-target. And of course, if condition allows, the impurity effects will be also studied, and the data should be also collected to support the future study.
Okay, understood. Thanks very much. Okay, and we just got another question here. What is GenScript's regulatory agency support track record in the US?
Oh, thanks for the question. So besides the 9 IND approvals globally in the presentation I mentioned before, we do expect 4 IND approvals in US this year and another one in 2025. And also, we are going to submit 1 MPD this year to MHRA in 2024. So this is mainly our plan this year and for the year after.
That's great news. Thanks very much for sharing. Okay, and last question I think we have for the moment. How does GenScript protect against possible cross-contamination? Are there any single-use policies for consumables in place?
Oh, sure. We do have a cross-contamination control strategy in place, and it is integrated in our quality management systems. And it is, in the design of the facility. For example, the manufacturing rooms are segregated with individual air handling units. And we do have the single-use policy for any product contacting consumables. For example, the tubes, the pipettes, and even the HPLC purification columns. Only they are in direct contact with the product, they will be single use only.
Okay, great. So quite a few safeguards in place then. Thanks so much for sharing. And thank you, Dr. Wong, for the very nice presentation today and also for taking the time to answer a few questions.
Yeah, thank you.
Okay, and thank you everyone again in the audience for joining us today. If you did submit a question, we didn't have time to get to it, we'll make sure to follow up with you via email as soon as we can. And thank you very much again for your time and attention today. And now I'll turn this back to XTalks for closing remarks.
Well, thank you very much for those answers, Dr. Wang and Scott, for moderating the Q&A. We've reached the end of the question and answer portion of this webinar. And if we couldn't attend to your questions, and there's a number of them out there, the team at GenScript will follow up with you after this presentation. Now, if you have any further questions, please direct them to the email address that you see there on your screen. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. Momentarily, I will share a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. So I encourage you to do that. Now, please join us in thanking our speaker, Dr. Wang, for his time here today. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now.